Okay, good morning. It just started pouring again outside of my office. It's really loud. It's kind of amazing how heavy the rain was last night and this morning. But I hope all of you are nice and dry, hopefully at home, or if you're on campus, you're actually somewhere where it's comfortable. Real quick, so this Friday will be our second article share day, and this will be the first time that I'm going to divide the two sections up. So the TIs, Alexis, and Zoe will be with the section 1 students, right? So those of you, the 7 of you who are not doing the writing intensive portion of this course, you'll be meeting with Alexis and Zoe. And I'm going to ask them, Alexis and Zoe, if you can go ahead today and post an announcement on the section 1 site with that different Zoom ID and password compared to our normal lecture one. And those students, you in section 1, keep an eye out for that information because that's what you'll use to log in on Friday for your article share. All right. Section 2 students, I think section 2 students, we can just continue to use the normal lecture login, right? There's no reason to have yet another third login involved here. Okay, so section 2 students, you just log in with the regular lecture stuff, or lecture ID and password. All right, so that's also a good reminder not to forget that tonight your article summary is due. Let's continue our discussion about tyrosine phosphorylation and also take a closer look at RAS pathways. Oh, so this is just another reminder then, right, that next week we have our first exam. So here we are in week 4. So here we are on Wednesday. Friday is going to be article discussion 2. I hope all of you remember to take the quiz. I did not send an extra reminder email, but hopefully you all did. Turn in your article summary assignment tonight. Then Friday we will meet for article share. On Monday we will have our last lecture of new material. That's our lecture 10. And then on Wednesday, we'll review during class time. And then Friday, we'll have the exam. And I'll talk more about the format of the exam on Wednesday, okay? So let's remind ourselves of some key concepts here. Normal cells follow environmental cues, for example, from other cells, as well as the types of proteins that are found in the extracellular matrix surrounding the cell. And these cues provide instructions that help influence things like proliferation, differentiation, apoptosis, and motility of the cell. So at any given time, a multitude of different signals are arriving at the surface of the cell and must be transduced into signaling pathways within the cell. Signaling pathways that involves proteins talking to other proteins, eventually leading to things like certain genes having their gene expression altered in the nucleus. So cancer then results from deregulation of molecules that participate in these pathways. Importantly, cancer is not from some newly invented message that the cell has never seen before. So that's a very important thing to think about. So let's illustrate this with sort of a little thought situation here, right? So if you think of a cell as a classical orchestra, right? So classical orchestra has all the different musicians and their instruments in it. Think about all the different musicians here as being proteins, right? And they are carrying out very specific contributions to the overall functioning of this orchestra's performance. Like proteins, they can be sort of grouped into different families like strings, brass, percussion, woodwinds. Right? And they all function together to play a piece of music in harmony, right? At least that's how it's supposed to go. So if you were to look at this example, right, you might say, the conductor here standing on the podium, is he the nucleus of the cell? Right? Because oftentimes you're tempted to think that because all the musicians are following what's going on by the conductor, right? But I would say no. I think a more intuitive way of thinking about this that helps you kind of understand the relationship of all these proteins working together, the function of the conductor, right, and the role that mutations play is by thinking about it this way. The musicians are proteins. The conductor himself is a protein, right? But the DNA is the sheet music. The DNA is the sheet music that each of the different instruments have in front of them, right? That give the instructions for what that particular protein's contribution should be to the cell. And when we say the performance of the musician interpreting the sheet music, that of course is the expression of the proper shaped protein and the ability of that properly shaped protein to carry out its job. The conductor then, right, is not really the nucleus, right? Because the conductor is actually just in control of controlling how all of the musicians are performing to make sure that everything is working together in harmony, right? So he is interpreting messages that aren't necessarily the specific notes, right? But interpreting signals, meaning things like dynamics, right? Louder, softer, or tempo, faster, slower, right? So the conductor then would be like those different types of receptors on the outside of the cell, like the EGFR growth factors or other types of mitogenic factors that interpret signals and use those to kind of control the activities of the different proteins, right? Whether those little proteins that are being controlled carry the functions out properly, right, can be influenced by the conductor being messed up, right, speeding up, slowing down when he shouldn't, whatever, right, or cutting things off when he shouldn't. But each protein, right, is really dependent upon their own set of sheet music in front of them, right, their own DNA that codes for the protein. Okay. So what is cancer then? Right? If you use this type of analogy, what is cancer? I think a lot of people, especially people, non-science types in the general public, would think cancer is like, all of a sudden, right? There's like a rock guitarist who's like crashing the orchestra, messing things up, or something that's new to the cell that doesn't belong in there, right? So this, I don't know how many of you recognize who this is, but this is Eddie Van Halen. And so I grew up like worshiping Eddie Van Halen, like me and my friends growing up outside Boston. It's so Boston because Boston's like stuck in the 80s. But anyhow, you know, we had our mullets and our denim jackets, and we all wanted to play guitar like Eddie Van Halen, right? Because he was awesome. You know, he just passed away last year, and so I was really sad about that. So I've been listening to a lot of Van Halen lately. Anyhow, so right. The point is, it's not like cancer is Eddie Van Halen suddenly coming in, playing his awesome guitar licks, and then all the orchestral musicians are freaking out and falling out of their chairs and stuff, right? No, right? Cancer is about deregulation of existing pathways, right? So what are we talking about here? Cancer might arise then from, for example, a particular musician no longer following the conductor, right? So if there's a loss of proper signal transduction taking place, it may be from the conductor himself or herself no longer interpreting things that are written in the score having to do with dynamics or tempo. It might be sheet music has been swapped between sections. So the sheet music for the violins is now being put in front of the trumpets, right? So that would be like chromosomal translocations. And obviously that would have a very deleterious impact on the harmony here. An individual player whose instrument is out of tune could be like a point mutation, right? Or a player who's playing way too loud would be like gene amplification. Right? So all of those types of things would mess up the harmony of this orchestra playing the piece. In some cases, it might be subtle. In some cases, it might be very dramatic. That is just like the types of changes that happen in a cell that transforms and becomes cancerous. So here's the orchestra again, right? But now we're looking at it in terms of what we've been discussing. So all of the blue proteins here are proteins that contribute in carrying out sort of their proper functions in order to keep harmony in the cell. They are all following the lead of these receptors, the receptors that interpret extracellular signals and tell different pathways, go faster, go slower, turn on, turn off, whatever, right? And then ultimately, these proteins here, they can have influence on each other, and they have influence on our genome in terms of then driving the expression of other genes. Okay, so if we think about that really complicated set of interactions on the previous slide, we can sort of simplify it down into sort of like this idea of flowcharts, right? Different signal pathways start from the top through different proteins interacting with different proteins, and importantly, right, certain pathways end up communicating with things like the nucleus and driving gene expression, right? As a function of different proteins interacting with each other along the way. So here's a question. What are some of the high priority genes that are actually being targeted here? If we said that many of the critical proto-oncogenes or proto-oncogenic pathways have to do with things like cell proliferation, driving the cell cycle forward, right? We've looked at examples of how those are controlled by things like receptor tyrosine kinases that are on the surface of the cell that interpret things like growth factors and send a signal through a chain of protein interactions into the cell to start encouraging things like moving forward in the cell cycle, right? So you may say, well, if we're interested in finding out what some of the most important sort of genes are in terms of pushing the cell cycle forward, right, obviously there's the receptors that are responsible for detecting that signal arriving in the first place, but you might want to know what are, let's say, the first wave of proteins that actually get turned on, or genes that get turned on, and a new wave of proteins that get made as sort of like the first wave of responding to growth factors arriving on the outside, right? So these early genes, as we call them, right, because these would be the ones that would first start being expressed and doing their job in response to growth factor, right? These were of particular interest to oncologists. So there are genes that get turned on right away, and then there are genes that, because those first genes got turned on, right, you have this weird sort of like trickling of other genes getting turned on because those first genes that came out in that first wave, those proteins start working, right? So very quickly, it gets to be kind of a messy situation, right? If you're watching a cell, you give it, like in cell culture, you starve the cell of any serum so it doesn't have any growth factors, so the cell is just kind of sitting there, right? That way you can have, a few million cells on a plate, all kind of like synchronized, right? Because at any given time, you have a few million cells, they're all going to be doing different things. Some of them are going to be dividing, some of them, whatever. You can synchronize this whole culture of cells by just removing serum from the media, right? So it's serum starved. So all the cells, after a day or so of not having any growth factors, they will all kind of be at the same point, right? Just sitting there. Then you can give these cells a big dose of serum and then look and see what happens, right? What new proteins start being made, let's say a few hours after that big dose of serum, and those are your early genes. But again, many of them are being made, right? The protein products will start feeding back and turning on other genes. So the reality is you're going to see a whole bunch of proteins. You cannot really tell, right, what were the first most important ones. So this is a very elegant experiment, classic experiment that was done. You take a bunch of cells, you serum starve them. And so this is just looking at one cell now. So the gray thing is the cell membrane, the plasma membrane, where you have something like an epidermal growth factor receptor, right? This orange thing here. And then this space in the middle would be like the cytoplasm. And then you have the nuclear membrane, right? So this brown thing would be the nuclear membrane. And then below this would be inside the nucleus now, right? So you serum starve them so that all the cells are kind of synchronized. So there's no growth factor binding to these receptors, and the cells are just kind of sitting there. So the intracellular levels of proliferation proteins then you would expect to be very low, right? You then go ahead and give them fresh serum, right? So now all of a sudden in the serum, you have this blue, blue diamond shape, right? It binds the growth factor receptor. The growth factor receptor then begins to signal. That can result in things like immediately changes in the cytoskeleton, right? We studied last time how certain types of receptors can actually change the cytoskeleton. It can also kick off a whole chain of protein-protein interactions that result in things like proteins being phosphorylated, right? So you have serine threonine kinases that are phosphorylating other targets, eventually leading to the phosphorylation of transcription factors, like this little green dot here, that then goes to work and starts binding to different promoters or enhancers around genes that would be considered the first wave of genes that the cell is going to now express as part of the cell cycle, now getting a little kick in the butt, and it's going to start moving, right? And so mRNAs are going to be made from those genes, and they're going to go out into the cytoplasm to then be translated, right? So the problem here is that some of those proteins, right, that are going to get translated are going to then go back into the nucleus and start turning on other waves of genes, right? And then the story gets a little messy. So the way you actually control this or limit this is by halting translation from taking place. So you can use compounds such as cyclohexamide. So cyclohexamide is a poison, right, that binds to ribosomes and prevents ribosomes from translating mRNAs into finished polypeptide chains, right? So you cannot make the finished protein product. Cyclohexamide actually comes from fungus. So this is one of the many poisons that are found in mushrooms. That's why many mushrooms are poisonous. They make stuff like this, right? Why do the mushrooms make it? Well, because it's a poison, right? To protect themselves so that deer and other animals that normally eat the mushrooms don't eat the brightly colored orange mushrooms, right? That are advertising that they have these poisons in them. But we can use cyclohexamide in the lab to do this. So you add cyclohexamide to these cells so that even though mRNAs are being made by that first wave of genes that are being turned on in response to growth factors, right? They don't get converted into finished proteins. Importantly, that means you don't have a buildup of proteins that's going to confuse things in here. Okay. Hi, Ben. You can, thanks for joining us. You can turn off your video, please. Okay, Ben, thanks. So that way you can just look at the production of mRNAs and very cleanly identify that if you see a buildup of mRNAs, right, these are all coming from these first wave immediate early genes that are being turned on. If you then did this experiment without the cyclohexamide, or you changed the media and took the cyclohexamide out and just put in full media again, right, that allows the ribosomes to go to work, you would make the protein products of those immediate early genes that were turned on, the yellow, orange, red, blue. Now you have the yellow, orange, red, blue proteins that are made, and they then may go back into the nucleus, many of them, and start turning on other genes, right? So this would be the second wave, and then there'd be a third wave, et cetera, right? And by then, the millions of cells on the plate would probably start getting out of sync from each other again, right? And they all start doing their own thing again. So what are some of these immediate early genes that researchers saw when they did this type of experiment? So this is a table, just a few examples of immediate early genes, right? So phos, Jun, things like MYC, right? So we already discussed that MYC is a very important transcription factor. And so you can see on the right here, it lists what type of protein it is. So these are transcription factors, TFs. Right. Many of these early genes are not transcription factors. They're not meant to go back and turn on other genes, but they're meant to just all of a sudden start changing things like the morphology of the cell as part of preparing for S phase and G2 and mitosis, right? So actin proteins suddenly get turned on to start remodeling the cytoplasm. Things like glucose transporters turn on, right? Because now all of a sudden the cell has been kickstarted. It's going to need to make a lot of proteins. So it's got to ramp up things like its cellular respiration, right? and things like cytokine proteins that are meant to be signaling molecules. Remember, we looked at on the previous lecture, you have epithelial cells, right, and supporting stromal cells. Right? They both make certain cytokines that the other type of cell has the receptor for, and that's how they talk back and forth. Right? Okay, so let's take a look. Let's see. We're going to talk about MYC a lot more later on, but I wanted to go back to this and focus on the other end of this pathway, right? Because we were just looking at how, okay, you can identify certain early genes that seem very important for the cell responding to mitogenic factors. Oh, so that term mitogenic means any type of factor that arrives on the cell that encourages it to head towards mitosis is mitogenic, right? So we talked about how they can identify certain first wave genes that are important for that. Let's go back to this end up here, right, the top part, and ask the question, how is it that proteins can specify which downstream partner they're supposed to talk to next? After all, if you look at in the background of this slide, there are other pathways that are happening, right, with other shapes. And you have a lot of crosstalk, things crossing over and stuff like that. How does the cell actually control which ones of these pathways needs to be kicked off, right? And how then you can properly turn on downstream proteins so that you really do get this kind of middle part here that achieves what you want it to achieve. And how do these pathways signal efficiency within a whole soup of proteins, right? Because just think about it this way. Even though these geometrical shapes are lined up neatly on this slide so that you can connect them with different lines, some of them are stimulatory lines, some of them are inhibitory lines. The reality is, in a cell, right, all these shapes are just floating around, bumping into each other, right? And so when we draw these things as a flowchart, that's purely just a functional way of us picturing it. In reality, the interactions are happening sort of at random, right? Things bumping into each other, causing the next step of a flowchart to take place. So how these receptors specify that very critical first downstream protein in the cytoplasm to activate has to do with the fact, again, that these pro-mitotic, pro-proliferative pathways, right, are the VIP pathways. They are the ones that are going to involve tyrosine phosphorylation. Remember, 99% of all the phosphorylation events that happen in your cell are serine threonines getting phosphorylated on proteins, right? Certain serines and threonines in proteins get phosphorylated. 99% of the stuff that's going on in your cell right now is that kind of phosphorylation. But tyrosine phosphorylation is reserved for these very important critical pathways that need to be tightly controlled. Right? So here we have two examples of receptor tyrosine kinases. In blue, we have the platelet-derived growth factor 1, or the PDGF receptor that we talked about previously. Here we have the red EGF receptor that we've talked about previously as well. Right? So these are different proteins, right? They're coded for by different genes. Their amino acid sequence is going to be different. Right? We already discussed how certain parts of their sequence are going to be tightly conserved, right? Because you're going to have these chunks of amino acid sequence that are the kinase domains, right? And these kinase domains end up phosphorylating threonines on the other tail of the partner for this. So remember, each one of these, in order for it to fully activate, there'd be two of these blue PDGF receptor molecules lined up, two of these red lined up, right? And the two tails of the red would be phosphorylating each other on tyrosines. The two tails of the blue one would be phosphorylating each other on the tyrosines, right? Here you see the positions of where the different tyrosines are in those tails. Right? So here, starting from amino acid position 579 through position 581, if you were to look at the amino acid sequence in this blue polypeptide chain, you'd see tyrosine, isoleucine, tyrosine, valine, right? So it's those two Ys, the two tyrosines, that can be phosphorylated by the other blue tail. right? And these are other highly conserved motifs, right? Motif just means any kind of sequence of anything that you see over and over again in evolution, right? It works very well, so therefore it appears in many different proteins, right? So these motifs you can see here, all of them involve the tyrosines, right? But they have different amino acids flanking them. And that's what gives them specificity, right? Because that means the shape of this region is going to be slightly different. Though the one here, the YIYV, is going to be a different three-dimensional shape than down here, YTAV, right? Because the amino acids are different. So this is where other downstream partners, the first cytoplasmic partners that are going to bind to these tails, come in. Right? So you can see here, you have a whole bunch of them over here that are important, including SRC, right? With the EGF receptor, you have a whole bunch of them, including things like SHC. All right, so we'll look at SHC in a couple slides. So let's focus just on SRC here and understand how it is that SRC knows to interact with this, okay, with this special sequence. So remember, SRC, we've looked at this before, is a protein that's made of several of these SRC homology domains. Remember, this cartoon shows that normally when SRC is inactive, it is up close to the cell membrane. It's anchored to the cell membrane on the inside, but it's kind of wrapped up. It's wrapped up because its C-terminal tail, a tyrosine on it, a tyrosine 530, is phosphorylated. That allows its SH2 domain to grab on to this phosphate group, right? And also this SH3 domain up here, you can tell just by looking at this, it's tightly bound to this other part of the SRC protein. And so the SRC protein is inactive, kept like this, right? If we look at, so this is the SH2 domain here, right? This is the shape over here. You notice that there are two alpha helices in this SH2 domain and this a little bit more liberally structured, beta sheet kind of looking thing in the middle. All right. So that's the secret here. This shape allows the SH2 domain of SRC to bind to specific phosphorylated tyrosines in the context of what other amino acids are surrounding those tyrosines. So let's take a closer look at this structure. Here it is. Again, here's the SH2 domain. So you have the two alpha helical parts of it in green and this little more unstructured kind of zigzaggy back and forth beta strand type of structure, right? The three-dimensional shape creates two pockets here. You can sort of visualize two pockets. So in those pockets are where phosphate groups that are on tyrosines can fit, right? Only if the amino acids that are surrounding those tyrosines also happen to fit in these grooves. So that's what gives SRC's SH2 domain specificity for binding this peptide stretch here that is a YIYV if those two Ys, those two tyrosines, are phosphorylated. Okay. Let's take a look at another region. Remember I said that SH3 region is bound up very tightly in the inactive SRC, right? And then once the SRC opens up, remember we studied last time, the way it opens up is that the phosphate from the tyrosine 530 gets removed, so it'll no longer fit into the SH2 domain, right? So the SH2 domain will let go, and that you can see now that 530, the C-terminal end, is now stuck out here. And that causes the protein to unravel. The SH3 then loosens its grip on whatever part of the protein it was holding onto before. And importantly, the SH3 is available to bind something else, right? So this SH3 domain, what does it bind? It binds proline-rich regions of proteins. So the colorful blue, green, gray is the SH3 domain of the SRC. The navy blue is another strand of some other peptide, in this case, a whole bunch of prolines, right? So you can see the rings sticking off. That is the right shape for the SH3 domain to wrap around it, right? So if we go back one slide, that suggests that this region of the SRC protein in yellow here itself is proline rich. That's why its own SH3 domain holds onto it so tightly as part of keeping the protein turned off, right? But once that proline-rich part of the SARC is no longer nearby because the whole SARC has opened up, this SH3 domain, it would love to bind some other thing that's proline-rich, right? And what do you think that is? Well, it turns out to be proline-rich stretches in the receptor tyrosine kinase, right? That are evolutionarily in the right spot, so that when you have the inactive SARC that then opens up, the SH2 domain here in blue can now happily bind to that phosphorylated tyrosines in the receptor tail here. So this is platelet-derived growth factor, right? And just upstream of that region of the tail, you have a proline-rich stretch that is positioned exactly the right spot where that SH3 domain of the SARC now happily grabs onto that, right? I think that's wonderful, right? Because this shows that it's all mediated by the tyrosines being phosphorylated in the first place on the tail that gets the SARC to go there. But now you have other sequences that encourage the SARC to really stabilize there, right? And then the SARC's activation domain then can then activate, right? And then SARC begins to kick off a whole series of other phosphorylation activities on downstream partners in the cytoplasm. All right, so that highlights the importance of conserved sequences being at the right spot, right? In this case, we're talking about the conserved motifs of amino acid sequences, right? Peptide sequences that can be used both by the binding of SARC, right? How SARC wants to find things onto the tail of the receptor tyrosine kinase. So these domains in SARC then are highly conserved, right? I already told you that's why the classic one is named after SARC itself. SARC homology domains, SH2, SH3, SH4, etc. And so these modules are used over and over again in different proteins. So many of them do appear in tyrosine kinases, right? So SARC is a tyrosine kinase, of course, right? So its next step then, once it activates, is it will then phosphorylate tyrosines on other proteins. But there's other ones as well. This is another example, FPS or SIC, right? They also utilize SH2 domains. So already you can imagine, right, that these will also stick to the tails of activated receptor tyrosine kinases, but they will selectively stick based upon whatever amino acids are variable in their SH2 domain to make their SH2 domain ever so slightly different than the SH2 domain of a different protein. So they will recognize only particular phosphorylated tyrosine patterns on the tail of the receptor. Some of these SH domains are used by other types of enzymes that are not kinases. So this is a GTPase activating protein, or a GAP, right? So you can see this GAP is made up of several of these SH domains and has a GAP catalytic domain. So what is a GTPase activating protein? Well, you take the name apart. It's going to activate a GTPase, right? And what's a GTPase? Well, it's some kind of an enzyme, because whatever this is, it's an ACE. And, you can already guess, some enzyme that catalyzes GTP, right? Maybe breaks the phosphate off, turning GTP to GDP, right? And you're like, oh, wait, we've heard that before. Where did we hear that? Okay, we'll talk about that. You have other ones like this one here, phospholipase C, that has SH3 domains, right? And it has groups that like to bind to lipid groups, right? Or more particularly, phospholipid groups, right? So where do you find phospholipids? You find them in the cell membrane, right? So already you can imagine whatever these PLC proteins do, they have something to do with modifying the cell membrane from the inside of the cell, working on the underside of the cell membrane, right, by its PLC domain. And you have other proteins that have no catalytic activity at all. They are nothing more than short little stretches of SH2 domains and SH3 domains, right? And so these are examples of GRAB2, for example, right? And you're like, well, what use is that, right? If it doesn't do anything except for have an SH2 and SH3 domain, so all it would be good at is sticking to certain motifs that are phosphorylated, right? And you're like, well, how is that helpful? Let's take a look. All right. All right. So when we talked about GTP being converted to GDP, hopefully some of you were like, yeah, I remember that has to do with RAS, right? RAS being switched on and off. So let's take a look at seeing what's going on here. So here's RAS, right? Remember, RAS is one of those supervisory proto-oncogenes. RAS is normally hanging out on the underside of the cell membrane, and it's usually bound to GDP. And in this state, RAS is in its off conformation, right? Remember, we said if the GDP gets swapped out and GTP gets put into RAS, right, that causes RAS to change its shape a little bit, and now RAS is active, and it can now signal downstream partners. So today, what we're talking about is what does that swapping of GDP to GTP? Right? The thing that does it is this guanosine exchange factor, or guanosine nucleotide exchange factor, or a GEF, right? So there's several examples of proteins that function as GEFs that exchange one guanosine nucleotide for another. In this particular case, the one that we're looking at in orange here is SOS. Right? If we go back a slide here, oh wait, SOS isn't even on here. Okay, but SOS is an example, right, of a guanine exchange factor that's going to encourage RAS to give up its GTP, and it'll give RAS a GTP instead, right? But the problem is this sauce would normally just be floating around somewhere else in the cell, right? Chances are it's not going to come anywhere near RAS. So sauce has to be recruited to be close to RAS if RAS is hanging out underneath the membrane here. How is it recruited? It's recruited by these SH2 domains and SH3 domains on that funny GRAB2 protein. So this is the one that I meant to go back for. Remember, GRAB2 at the bottom is just SH2 and SH3 domains. And we were sort of joking, what use is that? It acts as an adapter, right? A connection. And that's all it is, a physical connection between two other proteins that normally would not interact. So GRAB2, it has an SH2 domain that will bind to phosphorylated tyrosines on the tail of an activated receptor here, right? So the purple thing represents some generic tyrosine kinase, and it's a receptor, so it's an RTK, right? So that GRAB2 binds there, and with its SH3 domains, it binds to the proline-rich regions of SOS. So therefore, SOS is no longer floating around. It's now brought to the underside of the membrane, and guess who's waiting for it there? RAS. So this dramatically increases, right, hundreds of thousands of times more efficient interactions between SOS and RAS, and that's what gives RAS the GTP, right? So as long as RAS is hanging on to GTP, it is active. Right? But we said RAS is a GTPase. That means it can cleave GTP and turn back to GDP. So GTPase. On the previous slide, we said there is a protein known as GAP, right, which is a guanosine nucleotide activate or GTPase activating protein. So GAP is what encourages RAS to activate its GTPase activity, thereby having RAS convert the GTP back to GDP, and then RAS turns off again, right? So you can already see two different ways of controlling RAS activity here. Right? One is to activate RAS by exchanging its GDP for a GTP to turn it on. So that's done by SOS. And then another way is to shut RAS down by encouraging RAS to do its GTPase activity to convert GTP back to GDP. And that's facilitated by GAP. Again, because these proteins have SH2, SH3 domains, right, they may have been floating around in the cytoplasm, but once a receptor tyrosine kinase activates and has phosphorylated tyrosines on them, then these proteins then stick, right, and are then recruited to all be in the same spot, right? And that's a very crucial way of understanding how cells control different pathways. One of the conceptually, I think, most basic intuitive ways of controlling a pathway of proteins is just to encourage those participants, the proteins that are meant to be part of that pathway, to be close together so that interactions can happen between them, as opposed to just letting them float around in the cell, hoping that by chance they somehow bump into each other, right? So I think this is wonderful because this shows that that's how these receptor tyrosine kinases are so specific in controlling these very important pathways, right, through tyrosine phosphorylation, resulting in the recruitment of these proteins via their SH2, SH3 domains to act upon now and activate some of these higher level supervisor proto-oncogenes. All right. So once RAS activates, what happens? Okay. RAS itself, unlike SARC, is not a kinase. In fact, I'm still kind of unclear. This is kind of embarrassing to admit. I still don't have a great picture of how exactly RAS communicates with other downstream partners. My understanding is it's just a function of RAS changing its shape so that its three-dimensional surface is now the correct shape for other proteins that are floating by to fit. And then that encourages those proteins to change their shape, resulting in different downstream phosphorylation events to happen. Okay, those downstream phosphorylation events, though, from this point onward, right, are no longer tyrosine phosphorylation events. These will now be your run-of-the-mill, serine threonine phosphorylation events. Okay, so RAS signals three main downstream pathways that end up controlling things like proliferation, apoptosis, differentiation, cell motility. We're going to take a look at these one at a time. All right. Actually, we'll look at two of them in detail. I'll just mention the third. But let's look at this one first. One of the two very important ones is RAS talking to this MAPK pathway. All right. What does MAPK stand for? MAPK stands for mitogen activated protein kinases. Right. If you start from the bottom of the slide, these are examples of transcription factors. Many of these transcription factors, like ETS or ELK, were among those first wave immediate early genes that that start going to work. Okay, so the activation or expression of these genes, right, is important as one of the first responses to something like a mitogenic signal arriving on the outside, and in this case, it's going through RAS. So if you move one level up, right, the proteins that are going to control things like the expression or activation of these transcription factors are MAPKs, right? They are mitogen activated protein kinases that are going to phosphorylate other targets that ultimately end up regulating the activity of these transcription factors. So when researchers first discovered them, that made sense to call proteins that are at this level MAPKs, right? But then as they built the picture, the bigger picture of how these MAPKs are controlled, they realized, oh, these MAPK proteins themselves are activated by phosphorylation, right? That means there has to be another protein that acts as a kinase that phosphorylates them. And when they discovered this next higher up level of protein kinases, they came up with the very clever name of calling them MAPKs, right? So mitogen activated protein kinases, right? And you can see where this is going. They'd realized there's a higher level, proteins like RAF, right, which are the ones that phosphorylate the MAPKs. So they are called MAPKKs, right? I guess it's good in a way that they named it that way because this actually helps you keep straight where they should be in this chain, right? Okay, so I think that's it for today's lecture. Next time, we'll start off looking at another pathway that comes out of RAS, and that would be the PI3 AKT pathway. All right. And that will also be our introduction to another important group of proteins. Rather than the ones that are proto-oncogenes driving things forward, we also have a very important group of proteins for preventing cancer from happening. Those are the tumor suppressor proteins, right? So next lecture, when we look at the RAS PI3K pathway, we will also look at our first tumor suppressor. Okay, so I'll go ahead and stop